So Laura, please share your slides. I'm going to introduce you in the meantime. Laura Gribaldo is a medical doctor and holds a PhD in microbiology and virology. And she is a scientific officer at the European Commission's Joint Research Center, which is the European Commission's Science and Knowledge Hub. And she has 3 decades of experience in the field of testing for safety assessment. As a leader of the Applied Molecular Biology and Genomics Competence Group, she set up and managed a transcriptomics platform for the development of standardized assays in toxicology, and she has been responsible to support a program ensuring harmonization and validation of procedures in genetic testing for diagnostics purposes. And for the public health unit, she worked on rare diseases for a while and reported those to the European Union Committee on Experts on Rare Diseases. And today at the Chemical Safety and Alternative Methods Unit, she works in the field of knowledge dissemination, education, and training. And now she's going to present a project that was done at the JRC that she heads on human-based models in biomedical research. Laura, we are looking forward to your talk.
Okay. Thank you, Catherine. Can you hear me?
Yes.
Yes. And the slide is okay? The screen is okay?
Yes, looks good.
Okay, good. So thank you for the nice introduction, and thank you and the other organizer to invite me for joining these special events. Good morning, everybody from Italy. And today is raining, so the weather is not so nice like usual in Italy, but okay. I think that I prefer to switch my video because so that the audience can focus better on the slides. And since our work in the biomedical, sorry, I cannot. Okay, since our work in EGVAM in the biomedical field is relatively new in this area, I frequently open my presentation recalling the basis of our mandate, which are both the European legislation and the citizen engagement. And in fact, the Directive 2010, in respect to the former one, the 86/609, today has a wider scope and includes specifically animal use for the purposes of basic research, as well as for higher education and training. But also the citizen engagement is important as well, because the Stop Vivisection, which is the 3rd European Citizen Initiative submitted to the Commission on March of 2015, it was signed by more than 1 million of citizens in Europe. And the initiative originally requested the European Commission to abrogate the directive and to present a new proposal that does away with animal experimentation and makes compulsory the use of human data or data directly relevant for the human species. And the Commission was negative about the request of abrogate the directive, but it was positive to the request to engage more with all the stakeholders involved, all the scientific community involved in using animal models in research, in basic and applied research. And last but not least, what is most important, and it is behind the study that we started 1 year ago, is the last statistical report of the Commission on the use of laboratory animals in the years among the 2015-2017. As you can see from the graph, toxicity and other safety tests, including pharmacology, they accounted only for the 8% of the all animal uses, whereas the 70% of the animal are used in basic research and drug developments. And among them, the 92% are rodents, so mostly mice and rats. And another important aspect that was already mentioned by Elena, but it's important to stress the concept again, is that in the last decade, a number of national and international institutions founded a series of systematic review just to investigate the efficiency and the efficacy of animal models in understanding the human diseases of great impact on public health, like, as you see, respiratory disease, Alzheimer, as mentioned by Elena, but also autism disorder, liver disease, autoimmune disorder. So a series of very important human diseases. And in all the cases, these systematic review results published frequently in peer-reviewed journal, like for example this one in Drug Discovery Today, but many others. So the results show that the relevance of artificial animal model was considered overestimated in many cases. So some fundamental issues were overlooked, such as species-dependent differences, but also limitation of extrapolating data from animals to humans, as well as lack of validation of animal methods, or under-reporting of pitfalls and limitation of models, and also statistical underpowering of data. If we want to look also at some of the most used models in research, which are the transgenic mice, of course, they present advantages, and which is the reason why they are used. So they contain extra genetic material integrated into the genome. They carry oncogenes cloned for studying human cancer and so on. But if we look at the disadvantages on the scientific point of view, that there are and that are also important, like the fact that the integration is random, that many experiments reveal that the genetic surrounding of the inserted construct is modulating also the expression pattern of the transgenic self. And also in terms of time and cost, the transgenic rescue is time consuming, is expensive and labor intensive as well. Also for the knockout mouse, which is the other model frequently used, in parallel with some of advantages, here we see also the fact that 50% of these models are developmentally lethal. It's difficult to determine a gene function in relation to the human disease. And also in this case, the custom know-how mice is very expensive, still is very expensive. I wanted to summarize in a slide some common causes of reduced validity of animal studies. Some of them are considered many times and other not so much considered, but anyway, for example, the induction of the disease in young animals and otherwise healthy, whereas in patients the disease occurs in elderly people and frequently with comorbidities. The fact that the assessment of the effect of a treatment in an homogeneous group of animals versus of an heterogeneous group of patients. The gender, the use of either male or female animals only, whereas the disease occurs in male and female patients alike. The use of model for inducing a disease or injury with insufficient similarity to the human condition, and we would see this later. The delay to start of treatment that are unrealistic in the clinic, and the use of doses that are toxic or not tolerated by patients. And finally, differences in outcome measure and the timing of outcome assessment between animal studies and clinical trials. But reason of concerns are not only at the level of the biomedical approach per se, but even at the level of the basic science investigation. And for example, I mentioned in my presentation this paper published in Nature just last year. As is very interesting. It has been published by one of the main group of scientists in the world, the Allen Institute for Brain Science in Seattle, and they used single nucleus RNA sequencing to analyze the cell type present in the middle temporal gyrus of human cortex. And they identify a diverse set of excitatory and inhibitory neurons type, mostly sparse, and they compare this, the mouse cortex and the human cells cortex with the single cell RNA sequences. And despite the general conservation and homology among human and mouse cell type, they observed that at the level of the laminar distribution, at the level of gene expression, at the level of, especially of the behavior of the cells, there were big differences, huge differences. And so this species-specific feature emphasized the importance of directly studying human brain, and they conclude the study in Nature, recommending in future to the scientific community to shift from animal models to human-based models. So This is a need and a claim that is coming also from the scientific community today. So taking into account all these considerations, we decided to start with a study reviewing non-animal methods in use, already in use, available in basic and applied research. We apply some criteria to select the area of interest, and the criteria were the incidence and prevalence of human diseases, because we wanted to start from areas of impact on human health, on public health. Areas where most of the models are used for investigation of the mechanism of disease or drug discovery. And also areas where a potential causal link between chemical exposure and disease development were present. And for this reason, we decided to start from respiratory, the area of respiratory disease and the area of neurodegenerative diseases. The chronic respiratory diseases are one of the leading causes of morbidity and mortality globally. There are over 235 million of people affected by asthma, for example, and around 65 million that live with moderate to severe chronic obstructive pulmonary disease. And here, over the 90% of new drug programs fail to progress to the market, and the rate of successful transition from phase 2 to phase 3 clinical trial is very low, is 29%. If we consider the neurodegenerative disease, the problem is very similar. Alzheimer, as mentioned by Elena before, was one of the fastest rising disease among the leading 50 causes of global years of life lost between 1919 and 2013. And currently, there is no cure, no therapy for Alzheimer. Just some symptomatic treatments are available. And even in this area, the percentage of failure in drug development is very, very high, more than 90%, 95%, something like this. So we decided to launch this call for literature review in these two areas. And we got, at the end of the last year, the deliverables, which are a list of information sources, a categorized method list, which will be published in the JRC data catalog, 2 technical reports with a meta-analysis of these results, and the list of contacts in the field. And all this material, all this knowledge will be made publicly available on the EU Science Hub at our website, the URL EGVAM website, very soon. I want to show some preliminary results. When we had the first contact with the organizer of the event, I was asked to prepare a question for a poll, but there was no time eventually to show this question to the audience. The question was focusing on the idea we have about the number of already available non-animal models in basic research, because we were surprised at ourselves by the great number of non-animal models already published in literature in these two areas. For the respiratory tract disease, for example, we got 21,000 abstracts on non-cancer and cancer non-animal models, and we selected, applying a strict criteria of quality and many other criteria to select from these 21,000 of abstract the paper we wanted to analyze. We got 284 models for respiratory disease and 568 models for neurodegenerative disease. So, as you can see, quite a number of models already available in literature in these last 5 years, let's say. A list of models have been derived and characterized, and this table represents the way the results will be published soon. And it's a list of methods categorized on the basis of different categories. We have, for example, the disease feature. So you can see and you can look at the feature investigated through each model. It can be protein aggregation, inflammation. There are many features investigated. The type of the animal model, non-animal model used, the biological endpoint, so the direct marker of disease progression that is used in the model to describe the health effect can be DNA damage, protein dysfunction, and so on. The area of application of the model, which is also very interesting for our analysis. So if the model has been developed for drug testing or for the diagnostic purpose of a certain disease or for the study disease mechanism. The potential assay throughput of the model, which is the degree of automation of the experiments that can be done using this model, and so on. So if you, when you will enter this list of methods, you can go across the methods and you can select the ones of your interest depending on the, as I said, model type, disease feature, aim or application area, and so on. We also did already some statistics on these models to look at, for example, the number of methods identified for each disease area, and clearly lung cancer methods dominate in the area of respiratory disease, and followed by general disease model, which are not focusing on a specific disease, but more concerned with model development per se. But also the models for asthma are quite well represented. Whereas in the other areas, not so much. So there is room to improve in the other areas of disease. If we look at the area of application, the aim of the model development, most of these papers are on a model development per se, and are general exploratory methods, which are the most prominent across the different disease areas, and also models used for disease mechanism investigation. As you can see, the models developed for drug development and testing are less than the other models, and we strongly think that in this area of therapy development and drug testing, there is a huge need of non-animal models to be further developed and characterized and applied. And this is something that also is a message coming also from the pharmaceutical sector, which is very much interested in seeing new models developed for this purpose, for preclinical studies. If we look at the distribution of different models, for example, in Parkinson, we see that most of them are based on human or patient primary or stem cells, and human patient ex vivo tissues or body fluids. And I add also that for Parkinson, for example, most of the models are developed for looking at potential therapies. Whereas in Alzheimer, most of the models are developed for investigating the disease, because still we have to understand the mechanism behind this disease. And here we have a high percentage of model still based on human ex vivo tissues or body fluids, but also many models based on biochemical and cell-free assay. Some general relevant findings are that in the area of respiratory disease, the models still use a lot of stabilized cell lines. These are the most common one. Whereas for the neurodegenerative disease, most of the inventoried methods are based on induced pluripotent stem cells, so are quite advanced in respect to the area of respiratory disease. But for both the areas, the most innovative models, such as the 3D human tissue cultures, the spheroids, the organoids, the microfluidic system, are the most promising and relevant, but need to be further characterized. I like also to present some example, at least an example of an area of a disease where the application of non-animal model could be really of improvement for understanding the etiology and the pathology. behind the disease, and also to develop the proper therapies. This is the example of the idiopathic pulmonary fibrosis. Is a respiratory disease in which scars are formed in the lung tissues, resulting in stiffness, difficulty in breathing, which can eventually lead to respiratory failure, heart failure, and other complications. Is idiopathic, so refers to a manifestation without any known cause. But the current paradigm suggests that the alveolar epithelial cell damage is the key initiating event in this pathology. Today, the preclinical investigation of fibrosis use typically mouse models that are mouse models of bleomycin-induced pulmonary fibrosis, which are several limitations that prevent a direct translation to the human disease. So also the preclinical efficacy of currently approved antifibrotic agents is tested only in this bleomycin-induced model. And so this results in a therapy which is based on histological endpoints like, for example, collagen deposition, which is relevant in the mouse model, but it's not at all relevant clinically in humans. So you can imagine that in front of a study which is severe, very severe, as you can see, for the animals, absolutely not keeping in place the concept of animal welfare. But we have also problems of not efficiency for the human disease. So it's really something that it has to be changed. We can also add that reproducibility issues are evident because there is a variability in the experimental procedure between labs, leading to a lack of results, of consistent results, the animal science that need to be balanced with statistical power. So many factors that really tell us that it's time to move from these models to something better. If we look at the non-animal models, we have different models available here. For example, the preparation of human precision cut lung slices. These are humanized in vitro models that can really model the fibrotic disease using human cells. And these are, in theory, more suitable than animal model. In modeling human physiology, human biology, and immunity also. And this, for example, this technique recapitulates aspects of the human lung much better than other models. And so, therefore, the American Thoracic Society has recommended using this system to corroborate the results obtained with other models. So, future work should focus on developing antifibrotic drugs using humanized models and not the bleomycin administered animal models. We have other models available, suitable, for example, the pulmosphere that you see on the left side of the slide. The pulmosphere are 3D multicellular spheroid composed of lung cells from individual patients and also containing all the various cell types found in a human lung. And they simulate very well the microenvironment in the lung so that they can also serve for personalized medicine, personalized and predictive model, also for assessing responsiveness to drugs in the different patient that can respond in a different personalized way to the drugs. And also microfluidic co-culture offer opportunities to mimic a human disease model of pulmonary fibrosis. For example, in this case, let's say it's the model of pulmosphere and the precision cut slices at the moment are more advanced in respect to the microfluidic for the specific study of the pulmonary fibrosis. But I mean, it's important to make all this knowledge available, and then time by time, the scientists should evaluate which of these models is the most suitable one for the specific purpose of the aspect of the investigation that he or she is conducting. We also launched additional 5 review of non-animal models last year, and the deliverables are now getting ready. Very soon, they are arriving in these days. You see the different areas: cardiovascular disease, breast cancer, immunogenicity for advanced therapy medicinal products, autoimmune disease, and immuno-oncology models. I can anticipate you today that for the immunogenicity testing for advanced therapy medicinal products, we have 88 models available, collected, and most of these models were employed to test cell therapy products. We know that Europe is leading this area, the market of advanced therapeutics medicinal products. And from a biomedical perspective, one of the major bottlenecks is immunogenicity, because of the intrinsic property of this product of triggering the immune response in the patient, and so hindering the safety of the therapeutical strategy. And immunogenicity is especially important for cell-based products. The problem here is that we analyzing the first preliminary results in this area. We saw that the employment of the model was reporting for testing strategies of immunogenic response in allotransplantation or in mesenchymal stem cell application. Here, most of the time, T cells are used. T cells from peripheral blood of donors cultured in 2D condition are used to test immunogenicity. And there are very few studies reporting models like organoids and spheroids, so more advanced models. And there is a need to develop in this specific area something more advanced than T cells for peripheral blood as a model. And so this is something that we can recommend on this basis, that there is a room for innovation in this area. So looking for sustainable and well-characterized sources of human immune cells, as I said, but also to develop high throughput models, in silico models, and also specific technological platform for immunogenicity with high content assessment. And for example, a good initiative could be together test developer with the EMA working group on the application of 3Rs in regulatory testing. So try to really facilitate the dialogue among the different test developers, among the different scientists, to disseminate knowledge and to improve through the knowledge sharing this area of investigation. Who are the target user of this collection of non-animal models? There are many target users, we think. First of all, the research groups submitting a project proposal, the research group that makes use of living animals for their investigation, so the scientific community, but also the animal welfare bodies that have to advise the research group on the project proposals, and the competent authorities that are responsible for the project evaluation, and that have to say, yes, there is really a need to use animal for this project, or look, there are non-animal models available that can replace or can even be better than your animal models. So very important to also to them this collection of models, also to the national committee that coordinate this project evaluation, and for the national contact point in Europe who are responsible for the implementation of the directive in the member states. But finally, very important to me is that this collection will feed also another area of important interest for us, which is the area of education and training, because I think that this is the basis for promote properly alternative methods. So to inform the new generation of researchers on effective non-animal model currently used in biomedical research, so that in future they will be able to start to really a different reasoning when they have to conduct, when they have to design their experiment from the very beginning. So this is the state of the art of the moment. And if you have any question, you can make a direct question or even write to me to this email. Thank you.
Thanks very much, Laura. So now we are opening up the panel discussion with both speakers, with Helena and Laura. So please join me, both of you, switch on your microphones, and I will see what questions we got. Okay. So there are very specific, let's see.
They seem to be specific to me, some of them at least.
Yeah. Do you want to address the ones you can address quickly?
Yes, sure. So I think some of the questions is about the blood-brain barrier and our work, what we are doing, and how, as I also mentioned, it's one of the problem when it comes to drug delivery, for example, to the brain. The blood-brain barrier is very important. And one question is how we can our mini-brain model can be used to better understand the challenges to overcoming this challenge. So first, I will just mention our blood-brain barrier work. We have two different ways of doing it, one more successful than the other. Of course, the most simple, more successful, where we're just using inserts. So it's a quite common way of doing blood-brain barriers. We have endothelial cells from induced pluripotent stem cells on one side of this membrane, and then on the other compartment we have our mini brain. And then we are looking at how drugs or environmental toxicants can penetrate through this, and then we're measuring different things, the tear, for example, so the barrier for the blood-brain barrier, but we're also looking at how the brain sphere then, or the organoid, is affected by the compounds. So here we have used drugs or compounds that we know will pass the blood-brain barrier, and some that we know will not pass the blood-brain barrier. So it's pretty primitive, and it's more for us. We have mainly done it to do for virus infections. We're interested to see if the virus can really penetrate through the blood-brain barrier, which I think, sometimes that might not be possible. And the other thing is then, of course, for environmental toxicants, as I said, we're interested in toxicology. I don't think that our model is, in that sense, is the most prestige model to really try to answer some of those questions. However, there are a lot of very sophisticated blood-brain barrier models. One group I know from University of California, Chris Hughes, are doing some really exciting work, very fancy models. And we're actually talking about doing some collaboration there, but I'm happy to, if someone want to email me, I'm happy to share his presentation. And he has a company, Kino Biosensors. So I'm very happy to share that with you. So I said ours is a little bit more simple. One of the other questions was, I want to use lentiviral infection to knock down certain gene in cancer organoids. How can you assess the effect on cancer organoid? So I think one of the challenges, both in vitro and in vivo, is when you want to use some kind of lentiviruses, because the penetration into, if you want to do it within the organoid, can be challenging. We see it already when we do some immunohistochemistry. That is one of the challenges. Not all antibodies are penetrating to the core, so we don't always get a realistic picture from the core, through the whole organoid. What we are doing currently with lentiviruses is we're trying to create some reporter genes, and we're then doing this in a monolayer culture, actually in induced pluripotent stem cells, but you can probably do it at different stages as well. So that would, depending on what your question is, what you want to do with it, but might be easier to use first a monolayer culture and then culture them in 3D afterwards. And again, I think we're experts in toxicology, not really experts in either of these neurological disorders or in cancer. We just developed a tool, the mini brain, and felt that this could be useful in many other fields. So we are looking for collaborations, and some of the work I showed you have been collaborations. So if you have a project where you think the mini brain could be useful, please contact me. I would be very happy to discuss possibilities. We can teach you how to use it, but we need experts from outside that are experts in this neurological disorders and in cancer, for example, to really achieve something amazing with this. So we have the tool and we just need the application. So I hope I answered some. If you have more questions, please email me. I'm happy to talk to people offline also.
Helena, thanks a lot. I have a question for Laura.
Yes.
So I heard that you plan to build a database. What is the idea for disseminating the information? Because obviously this is very important, because we're talking about quality of science here. And I think we can all agree that if the animal model is not a valid model, we have to find something else. It doesn't help that we refine models in terms of animal welfare if they are not translatable to the human setting. And you showed that very well in your presentation, that there are a number of animal models that don't seem to be suitable. So what is the idea? Because obviously not only hopefully the European Union will benefit from your research, but what are your ideas for that?
Yeah, thank you. I can also reply with the same answer to you and also to Orla, who asked about the date, or when this data will be available and how. So first of all, the reason why we waited a bit before publishing these results was that we wanted to wait first the results of the statistical report of the Commission, just to be able to cross-check the area of major animal consumption and if they were, fitting well with our results. So we waited till February for the publication of the statistical report, and now we had to work on the recommendation for antibody production. So this is the reason why the results were a bit postponed, let's say. But we are going to publish the list of the method, the inventory of the method and the technical report by the end of June, hopefully. They are ready there. So, but apart of making them available on our platform, I think that what is important now, and we are discussing a lot with our hierarchy and our colleague, is not simply the dissemination of this information, because the important thing is to improve the knowledge sharing, which is different from the dissemination per se. It means that we have to make some efforts in understanding which will be the best platform to improve the dialogue among the scientists. At the end of the day, I think that first of all, we want the scientific community be able to discuss openly on a common platform of these models, the pros and cons of these different models they are using, because I think they are the ones that know where is better to use one method than another. So we want really to create something on knowledge sharing. So package this information in the best way, but also try to develop this sort of dialogue through events, of course, through workshops or with these experts, but also, if possible, creating some informatic platform or a place where it's possible to have this talk, this dialogue. And then, as I said, also another way of dissemination of knowledge sharing is through education and training programs. So training for trainers and training for universities, not only the summer school that we are going to organize for the next year, but also through the collaboration with the university networks worldwide and in Europe. So we would like really to take different targets and be able to outreach these different targets. So at the level of education, universities, and so on. As I said, national and ethical committees, and to package this information and this knowledge in a different way for these different target people. But the most important ones, of course, are the scientists, and we want to target them. Let's see if it's feasible, this idea we had to yeah, to start with a platform of dialogue among them. Yes, that's the way. And we think that it's important that we find a way also to, yeah, to understand how to qualify these methods. For example, one request that comes from the pharmaceutical sector is start a discussion on the qualification of this method, because they are very much interested in qualified methods for drug development. So it's a lot of work, but we have some idea to to develop on. And the last, there was a question from Norecopa, from Dr. Smith. He mentioned that there are 2 inventories already published in Norecopa, but I know them, but they are not the inventories on non-animal models. They are inventories on knowledge sources, one, and the other one is an inventory of maybe teaching tools or something like this. Yeah. Okay.
Thank you very much. Yeah, I think, yeah, the question is, you know, how do we approach animal experimenters who are used to using animal models? And I mean, there's more and more data coming out, systematic reviews, citation analyses, meta-analyses that show that we have a translation problem, and that it's not, in most cases, not going to be enough to just improve the models, because we have...
Of course, it's also important funding. It's important also to lobby with our colleagues at the DGRTD in Brussels, and be able in a short future to focus funding on these non-animal models. So
Yeah.
to be able to publish a number of calls for funding for project specifically based on non-animal models in these areas of disease.
Yeah, yeah. We have another question asking, since these artificial models like the complex architecture and interaction of live evolved animal models, would you expect poorer translatability to human health and safety for many or most applications? I mean, I don't think, I want you to hear your take, but it's not artificial models, it's human biology based models. I mean, they seem artificial because it's only, you know, it's a part of the human basically, but maybe you can elaborate on this.
Yes, indeed, they are not artificial models. So there is now also a great area of interest based on artificial intelligence and you know, modeling, computational modeling, and mathematical model. The ones that we mentioned are specifically biological based models. So they are really mimicking the architecture of the life in the tissue of the bodies. So I mean, they are not, as you said, they are not artificial at all. Of course, all these systems, they are not per se a whole body like the body of the animal, but we saw through the systematic review, through many evidences, that these whole animal models are not at all predicting what is happening in humans. So I mean, I think that at the limit, what they do is to cover different pieces of a mechanism, and they do this in a way that is not better than the one that is done by the human-based model. So I think that yes, we have, and sometimes, let's say, we have to be clear. Sometimes still the animal models is not avoidable because of some reasons, because we don't have available models in specific areas and so on. But frequently what happens is that, and this is something that I have as a feedback from the scientists themselves, that sometimes the scientists, they complement their non-animal model with the animal ones, because there is a request, for example, from the editorial policies of the journals, because sometimes they could, you know, finalize the study avoiding animal models, but they have to do the experiments in the animals. because of this request coming from the journals. So this is another area where we should be more active in lobbying with these journals and see what is possible to promote at the level of the editorial policies.
Yeah, I think Helena can also speak to that. Obviously, we are not conducting animal experiments at our center, and sometimes that's a request which is explainable why it's not scientifically necessary. Helena, I think there's another question for you.
Yes, yes, I saw that. So how would the effects of drugs on hormones and vice versa be studied using the brain organoid model without using animal models? It's a very good question, I have to say, and we have been quite ignorant about the hormone system, not specifically for us, but for all people that are modeling things. The same with, using in vitro models where other things we are lacking as well, metabolism, for example. But one of the way going forward now is to use the human on chip approach to really try to combine different organs as well. So you have these three-dimensional complex organs with the hormonal system. So there are some very nice reproductive systems, different organs of the reproductive system, the female reproductive system available. And there's also, of course, a lot of liver models. So one way is combining that. I am also involved in an ADAST advisory scientific board for Endpoint, which is one of the Horizon 2020 on the European side bigger projects, where we're looking at endocrine disruption and developmental neurotoxicity, which we know already, like thyroid interruptions of the thyroid hormone, for example, has an effect of the development of the brain. And we're now trying to address this, or they're trying in this project to address this using only in vitro systems. And they have started to talk about ways of also modeling the hormone system within the media compartment then. So simplified again, but this is areas, I don't say that everything can be modeled in vitro at this point. I am sure it cannot, but I think we are getting there, and I think being more creative, more things coming up, then I think we might get there eventually. But sometimes the animal, as Laura says, will be the best model and will be important. But if we can reduce it at least and just use it when it's necessary, and also use it in the right settings. So we should know what species to look at, for example. We shouldn't just, try to create more transgenic animals because I'm not sure that will make us going forward, because there's so many things you need to change to make a mouse a human. So that's my 2 cents on this.
Yeah, thank you very much. I think that was a good last point. To just say we need a discussion about the models, and we need to really be honest about their benefits and their limitations of all models, animal models as well as non-animal models. And so the work that JRC has been doing will be very, very important and interesting for us to look at, and hopefully there will be a big discussion in the research community about that, and it will be a way forward to leave behind some of the models that haven't really served us and haven't really improved human health care. So now we are turning away from replacement. Thank you again to Helena and Laura for their presentations.